Hi there, let's begin with this super interesting chapter. While examining a thin slice of cork, Robert Hooke saw that the cork resembled the structure of a honeycomb, consisting of many little compartments. Cork is a substance which comes from the bark of a tree. This was in the year 1665 when Hooke made this chance observation through a self-designed microscope. Robert Hooke called these boxes cells. Cell is a Latin word for a little room. This may seem to be a very small and insignificant incident, but it is very important in the history of science. This was the very first time that someone had observed that living things appear to consist of separate units. The use of the word cell to describe these units is being used till this day in biology. Let us find out about cells. Leeuwenhoek, in 1674, with the improved microscope, discovered the free-living cells in pond water for the first time. It was Robert Brown in 1831 who discovered the nucleus in the cell. Purkinje in 1839 coined the term protoplasm for the fluid substance of the cell. What are living organisms made up of? Let's perform an activity. Peel off an onion skin from the inner side of onion with the help of forceps and put it on a glass slide. Add a drop of saffron solution and put a cover slip on it, avoiding any air bubbles. Now observe it under a low power microscope and then a high power microscope. What are these structures? These structures look similar to each other. Together they form a big structure like an onion bulb. In fact, onion bulbs of different sizes have similar small structures visible under a microscope. The cells of the onion peel will all look the same, regardless of the size of the onion they came from. These small structures that we see are the basic building units of the onion bulb. These structures are called cells. Not only onions, but all organisms that we observe around are made up of cells. Now one question here. What do you think? When we put solution on the peel, did each cell get evenly colored? Well, according to their chemical composition, different regions of the cells get colored differentially. Some regions appear darker than the other regions. Now, why is the cell called the structural and functional unit of life? The cell is called so because all living organisms are made up of cells. Cells are also essential for performing various life processes required for sustaining life. Furthermore, cells provide form and structure, process nutrients and convert them into usable energy. The invention of magnifying lenses led to the discovery of the microscopic world. It is now known that a single cell may constitute a whole organism that lives on its own, as in amoeba, chlamydomonas, paramecium, and bacteria. These organisms are called unicellular organisms because uni means single. On the other hand, many cells group together in a single body and assume different functions in it to form various body parts in multicellular organisms, such as some fungi, plants, and animals. Every multicellular organism has come from a single cell. How? Cells divide to produce cells of their own kind. All cells thus come from pre-existing cells. Some organisms can also have cells of different kinds. Here are some cells from the human body: smooth muscle cell, blood cells, nerve cell, bone cell, ovum, sperm, fat cell, etc. The shape and size of cells are related to the specific function they perform. In some cases, the cell shape could be more or less fixed and peculiar for a particular type of cell. For example, nerve cells have a typical shape. Some cells, like amoeba, have changing shapes. The cell theory that all the plants and animals are composed of cells and that the cell is the basic unit of life was presented by two biologists, Schleiden and Schwann. The cell theory was further expanded by Virchow in 1855 by suggesting that all cells arise from pre-existing cells. With the discovery of the electron microscope in 1940, it was possible to observe and understand the complex structure of the cell and its various organelles. Each living cell has the capacity to perform certain basic functions that are characteristic of all living forms. How does a living cell perform these basic functions? We know that there is a division of labor in multicellular organisms such as human beings. This means that different parts of the human body perform different functions. The human body has a heart to pump blood, a stomach to digest food, and so on. Similarly, division of labor is also seen within a single cell. In fact, each such cell has got specific components within its own, known as cell organelles. Each kind of cell organelle perform a special function, such as making new material in the cell, clearing up the waste material from the cell, and so on. A cell is able to live and perform all its functions because of these organelles. These organelles together constitute the basic unit called the cell. It is interesting that all cells are found to have the same organelles, no matter what their function is or what organism they are found in. Hope you're liking the video. In that case, please hit the like button and subscribe to the channel. What is a cell made up of? What is the structural organization of a cell? Let's see how a cell is organized. If we study a cell under a microscope, we would come across three features in almost every cell: plasma membrane, nucleus, and cytoplasm. All activities inside the cell and interactions of the cell with its environment are possible due to these features. Let us see how. Plasma membrane or cell membrane. This is the outermost covering of the cell that separates the contents of the cell from its external environment. The plasma membrane allows or permits the entry and exit of some materials in and out of the cell. It also prevents movement of some other materials. The cell membrane, therefore, is called selectively permeable membrane. Now the question is, how does the movement of substances take place into the cell? How do substances move out of the cell? Well, some substances like the carbon dioxide or oxygen can move across the cell membrane by a process called diffusion. During diffusion, there is spontaneous movement of a substance from a region of high concentration to a region where the concentration is low. Something similar to this happens in cells when, for example, carbon dioxide is a cellular waste and requires to be excreted out by the cell. It accumulates in high concentrations inside the cell. In the cell's external environment, the concentration of carbon dioxide is low as compared to that inside the cell. As soon as there is a difference of concentration of carbon dioxide inside and outside a cell, carbon dioxide moves out of the cell from a region of high concentration to a region of low concentration outside the cell by the process of diffusion. Similarly, oxygen enters the cell by the process of diffusion when the level or concentration of oxygen inside the cell decreases. Thus, diffusion plays an important role in gaseous exchange between the cells as well as the cell and its external environment. Water also obeys the law of diffusion. The movement of water molecules through such a selectively permeable membrane is called osmosis. The movement of water across the plasma membrane is also affected by the amount of substance dissolved in water. Thus, osmosis is the passage of water from a region of high water concentration through a selectively permeable membrane to a region of low water concentration till equilibrium is reached. What will happen if we put an animal cell or a plant cell into a sugar solution or salt solution in water? One of the three things could happen. If the medium surrounding the cell has a higher water concentration than the cell, meaning that the outside solution is very dilute, the cell will gain water by osmosis. Such a solution is known as hypotonic solution. Water molecules are free to pass across the cell membrane in both directions, but more water will come into the cell than will leave. The net result is that water enters the cell. The cell is likely to swell up. If the medium has exactly the same water concentration as the cell, there will be no net movement of water across the cell membrane. Such a solution is known as isotonic solution. Water crosses the cell membrane in both directions, but the amount going in is the same as the amount going out, so there is no overall movement of water. The cell will stay the same in size. If the medium has a lower concentration of water than the cell, meaning that it is a very concentrated solution, the cell will lose water by osmosis. Such a solution is known as a hypertonic solution. Again, water crosses the cell membrane in both directions, but this time more water leaves the cell than enters it. Therefore, the cell will shrink. Thus, osmosis is a special case of diffusion through a selectively permeable membrane. Here are a couple of activities that can be tried for osmosis. Osmosis with an egg. Remove the shell of an egg by dissolving it in dilute hydrochloric acid. The shell is mostly calcium carbonate. A thin outer skin now encloses the egg. Put the egg in pure water and observe after 5 minutes. What do we observe? The egg swells because water passes into it by osmosis. Now place a similar deshelled egg in a concentrated salt solution and observe for 5 minutes. The egg shrinks. Why? Because water passes out of the egg solution into the salt solution because the salt solution is more concentrated. We can also try a similar activity with dried raisins or apricots. Put dried raisins or apricots in plain water and leave them for some time. Then place them into a concentrated solution of sugar or salt. You will observe the following. Each gains water and swells when placed in water. However, when placed in concentrated solution, it loses water and consequently shrinks. Unicellular freshwater organisms and most plant cells tend to gain water through osmosis. Absorption of water by plant roots is also an example of osmosis. Thus, diffusion is important in exchange of gases and water in the life of a cell. In addition to this, the cell also obtains nutrition from its environment. Different molecules move in and out of the cell through a type of transport requiring use of energy. The plasma membrane is flexible and is made up of organic molecules called lipids and proteins. However, we can observe the structure of the plasma membrane only through an electron microscope. The flexibility of the cell membrane also enables the cell to engulf in food and other materials from its external environment. Such processes are known as endocytosis. Amoeba acquires its food through such processes. What is an electron microscope? An electron microscope is a microscope that uses a beam of accelerated electrons as a source of illumination. Cell wall. Plant cells, in addition to the plasma membrane, have another rigid outer covering called the cell wall. The cell wall lies outside the plasma membrane. The plant cell wall is mainly composed of cellulose. Cellulose is a complex substance and provides structural strength to plants. When a living plant cell loses water through osmosis, there is shrinkage or contraction of the contents of the cell away from the cell wall. This phenomena is called plasmolysis. We can observe this phenomena through an activity. Mount the peel of Rio leaf in water on a slide and examine cells under the high power of a microscope. Note the small green granules called chloroplasts. These chloroplasts contain a green substance called chlorophyll. Put a strong solution of sugar or salt on the mounted leaf of the slide. Wait for a minute and observe under a microscope. What do we see? Now place some Rio leaves in boiling water for a few minutes. This kills the cells. Then mount one leaf on the slide and observe it under a microscope. Put a strong solution of sugar or salt on the mounted leaf of the slide. Wait for a minute and observe it again. What do we find? Did plasmolysis occur now? What do we infer from this activity? It appears that only living cells and not dead cells are capable of absorbing water by osmosis. And cell walls permit the cells of plants, fungi, and bacteria to withstand very dilute, that is, hypotonic external media without bursting. In such media, the cells tend to take up water by osmosis. The cell swells, building up pressure against the cell wall. The wall exerts an equal pressure against the swollen cell. Because of their walls, such cells can withstand much greater changes in the surrounding medium than animal cells. Let's perform an activity to observe cells from our own body. Let us take a glass slide with a drop of water on it. Using an ice cream scoop, gently scrape the inside surface of the cheek. Does any material get stuck on the spoon? With the help of a needle, we can transfer this material and spread it evenly on the glass slide kept ready for this. To color the material, we can put a drop of methylene blue solution on it. Put a cover slip on it. Now the material is ready for observation under microscope. Darkly colored spherical or oval dot-like structure near the center of each cell is called nucleus. The nucleus has a double-layered covering called nuclear membrane. The nuclear membrane has pores which allow the transfer of material from inside the nucleus to its outside, that is, to cytoplasm. The nucleus contains chromosomes, which are visible as rod shaped structures only when the cell is about to divide. Chromosomes contain information for inheritance of characters from parents to next generation in the form of DNA or deoxyribonucleic acid molecules. Chromosomes are composed of DNA and protein. DNA molecules contain the information necessary for constructing and organizing cells. Functional segments of DNA are called genes. In a cell which is not dividing, this DNA is present as part of chromatin material. Chromatin material is visible as entangled mass of thread-like structures. Whenever the cell is about to divide, the chromatin material gets organized into chromosomes. The nucleus plays a central role in cellular reproduction, the process by which a single cell divides and forms two new cells. It also plays a crucial part along with the environment in determining the way the cell will develop and what form it will exhibit at maturity by directing the chemical activities of the cell. In some organisms like bacteria, the nuclear region of the cell may be poorly defined due to the absence of a nuclear membrane. Such an undefined nuclear region containing only nucleic acids is called a nucleoid. Such organisms whose cells lack a nuclear membrane are called prokaryotes. Pro means primitive or primary, and karyote means nucleus. Organisms with cells having a nuclear membrane are called eukaryotes. Prokaryotic cells also lack most of other cytoplasmic organelles present in eukaryotic cells. Many of the functions of such organelles are also performed by poorly organized parts of the cytoplasm. The chlorophyll in photosynthetic prokaryotic bacteria is associated with membranous vesicles, which are bag-like structures, but not with plastids as in eukaryotic cells. Cytoplasm. A large region of each cell is enclosed by the cell membrane. This region is called the cytoplasm. It takes up very little stain. The cytoplasm is the fluid content inside the plasma membrane. It also contains many specialized cell organelles. Each of these organelles performs a specific function for the cell. Cell organelles are enclosed by membranes. In prokaryotes, besides the absence of a defined nuclear region, the membrane-bound cell organelles are also absent. On the other hand, the eukaryotic cells have nuclear membrane as well as membrane-enclosed organelles. The significance of membranes can be illustrated with the example of viruses. Viruses lack any membranes and hence do not show characteristics of life until they enter a living body and uses its cell machinery to multiply. Let's see some facts about prokaryotic cells and eukaryotic cells. A prokaryotic cell is generally 1 to 10 micrometer, whereas a eukaryotic cell is generally large, between 5 to 100 micrometer. Nuclear region in eukaryotic cell is well defined and surrounded by a nuclear membrane. Let us know in the comment section about the nuclear region in prokaryotic cell. While prokaryotic cell has a single chromosome, there are multiple in a eukaryotic cell. Membrane-bound cell organelles are absent in prokaryotic cell. How about the eukaryotic cell? Let us know in the comment section below. Cell organelles. Every cell has a membrane around it to keep its own contents separate from the external environment. Large and complex cells, including cells from multicellular organisms, need a lot of chemical activities to support their complicated structure and function. To keep these activities of different kinds separate from each other, These cells use membrane-bound little structure or organelles within themselves. This is one of the features of the eukaryotic cells that distinguish them from the prokaryotic cells. Some of these organelles are visible only with an electron microscope. Some important examples of cell organelles are endoplasmic reticulum, Golgi apparatus, lysosomes, mitochondria, and plastids. They are important because they carry out some very crucial functions in cells. Hope you're liking the video. Don't forget to subscribe and like. Endoplasmic reticulum. The endoplasmic reticulum is a large network of membrane-bound tubes and sheaths. It looks like long tubules or round or oblong bags called vesicles. The ER membrane is similar in structure to the plasma membrane. There are two types of endoplasmic reticulum: rough endoplasmic reticulum and smooth endoplasmic reticulum. RER or rough endoplasmic reticulum looks rough under a microscope because it has particles called ribosomes attached to its surface. The ribosomes, which are present in all active cells, are the sites of protein manufacture. The manufactured proteins are then sent to various places in the cell depending on the need, using the endoplasmic reticulum. The smooth endoplasmic reticulum helps in the manufacture of fat molecules or lipids, important for cell function. Some of these proteins and lipids help in building the cell membrane. This process is known as membrane biogenesis. Some other proteins and lipids function as enzymes and hormones. Although the ER varies greatly in appearance in different cells, it always forms a network system. Thus, one function of the endoplasmic reticulum is to serve as channels for the transport of materials, especially proteins, between various regions of the cytoplasm or between the cytoplasm and the nucleus. The endoplasmic reticulum also functions as a cytoplasmic framework, providing a surface for some of the biochemical activities of the cell. In the liver cells of the group of animals called vertebrates, SER plays a crucial role in detoxifying many poisons and drugs. Golgi apparatus. The Golgi apparatus, first described by Chemio Golgi, consists of a system of membrane-bound vesicles, which are flattened sacs arranged approximately parallel to each other in stacks called the cisterns. These membranes often have connections with the membranes of endoplasmic reticulum and therefore constitute another portion of a complex cellular membrane system. The material synthesized near the endoplasmic reticulum is packaged and dispatched to various targets inside and outside the cell through the Golgi apparatus. Its functions include the storage, modification, and packaging of products in vesicles. In some cases, the complex sugars may be made from simple sugars in the Golgi apparatus. The Golgi apparatus is also involved in the formation of lysosomes. Chemio Golgi was born at Corteno near Brescia in 1843. He studied medicine at the University of Pavia. After graduating in 1865, he continued to work in Pavia at the Hospital of St. Matteo. At that time, most of his investigations were concerned with the nervous system. In 1872, he accepted the post of Chief Medical Officer at the Hospital for the Chronically Sick at Abbiategrasso. He first started his investigations into the nervous system in a little kitchen of his hospital, which he had converted into a laboratory. However, work of greatest importance, which Golgi carried out, was a revolutionary method of staining individual nerve and cell structures. This method is referred to as black reaction. This method uses a weak solution of silver nitrate and is particularly valuable in tracing the processes and most delicate ramifications of cells. All through his life, he continued to work on these lines, modifying and improving this technique. Golgi received the highest honors and awards in recognition of his work. He shared the Nobel Prize in 1906 with Santiago Ramon Cajal for their work on the structure of the nervous system. Lysosomes. Structurally, lysosomes are membrane-bound sacs filled with digestive enzymes. These enzymes are made by rough endoplasmic reticulum. Lysosomes are a kind of waste disposal system of the cell. These help to keep the cell clean by digesting any foreign material as well as worn-out cell organelles. Foreign materials entering the cell, such as bacteria or food, as well as old organelles, end up in the lysosomes, which break complex substances into simpler substances. Lysosomes are able to do this because they contain powerful digestive enzymes capable of breaking down all organic material. During the disturbance in cellular metabolism, for example, when the cell gets damaged, lysosomes may burst and the enzymes digest their own cell. Therefore, lysosomes are also known as the suicide bags of a cell. Mitochondria. Mitochondria are known as the powerhouses of the cell. Mitochondria have two membrane coverings. The outer membrane is porous, while the inner membrane is deeply folded. These folds increase surface area for ATP generating chemical reactions. The energy required for various chemical activities needed for life is released by mitochondria in the form of ATP or adenosine triphosphate molecules. ATP is known as energy currency of the cell. The body uses energy stored in ATP for making new chemical compounds and for mechanical work. Mitochondria are strange organelles in the sense that they have their own DNA and ribosomes. Therefore, mitochondria are able to make some of their own proteins. Plastids. Plastids are present only in plant cells. There are two types of plastids: chromoplasts or the colored plastids, and leucoplasts or the white or colorless plastids. Chromoplasts containing the pigment chlorophyll are known as chloroplasts. Chloroplasts are important for photosynthesis in plants. Chloroplasts also contain various yellow or orange pigments in addition to chlorophyll. Leucoplasts are primarily organelles in which materials such as starch, oils, and protein granules are stored. The internal organization of the chloroplast consists of numerous membrane layers embedded in a material called the stroma. These are similar to mitochondria in external structure. Like the mitochondria, plastids also have their own DNA and ribosomes. Vacuoles. Vacuoles are storage sacs for solid or liquid contents. Vacuoles are small sized in animal cells, while plant cells have very large vacuoles. The central vacuole of some plant cells may occupy 50 to 90% of the cell volume. In plant cells, vacuoles are full of cell sap and provide turgidity and rigidity to the cell. Many substances of importance in the life of plant cell are stored in vacuoles. These include amino acids, sugars, various organic acids, and some proteins. In single-celled organisms like amoeba, the food vacuole contains the food items that the amoeba has consumed. In some unicellular organisms, specialized vacuoles also play important roles in expelling excess water and some waste from the cell. Each cell thus acquires its structure and ability to function because of the organization of its membrane and organelles in specific ways. The cell thus has a basic structural organization. This helps the cells to perform functions like respiration, obtaining nutrition, and clearing of the waste material or forming new proteins. Thus, the cell is the fundamental structural unit of living organisms. It is also the basic functional unit of life. Here are the parts of animal cells and plant cells which we just discussed. Let's do a short recap, after which we would cover our last topic, cell division. Can you name the two organelles we have studied that contain their own genetic material? Mitochondria and plastids. If the organization of a cell is destroyed due to some physical or chemical influence, what will happen? The cell will not be able to perform its basic functions like respiration, nutrition, excretion. This may stop all the life activities inside the cell, eventually leading to its own death. Why are lysosomes known as suicide bags? During the disturbance in cellular metabolism, for example, when the cell gets damaged, lysosomes may burst and the enzymes digest their own cell. Where are proteins synthesized inside the cell? The ribosomes are transiently connected to the rough endoplasmic reticulum where the proteins are produced. Cell division. New cells are formed in organisms in order to grow, replace old, dead, and injured cells, and to form gametes required for reproduction. The process by which new cells are made is called cell division. There are two main types of cell division: mitosis and meiosis. The process of cell division by which most of the cells divide for growth is called mitosis. In this process, each cell called mother cell divides to form two identical daughter cells. The daughter cells have the same number of chromosomes as mother cell. It helps in growth and repair of tissues in organisms. Specific cells of reproductive organs or tissues in animals and plants divide to form gametes, which after fertilization give rise to offspring. They divide by a different process called meiosis, which involves two consecutive divisions. When a cell divides by meiosis, it produces 4 new cells instead of just 2. The new cells only have half the number of chromosomes than that of the mother cells. Can you think as to why the chromosome number has reduced to half in daughter cells? Well, this is because meiosis creates cells that are destined to become gametes or reproductive cells. This reduction in chromosome number is critical. Without it, the union of two gametes during fertilization would result in offspring with twice the number of chromosomes. With this interesting chapter comes to an end. We would come back with some more. Till then, keep watching ISA Kids. Don't forget to hit the like, share, and subscribe button. Have a great day. Bye bye.